And the timing of tonight's session is excellent. As I'm sure you're all aware, we're in MND week, so we're probably feeling especially focused on the impacts of MND. So just before we start, I'd like to have an acknowledgement of country first. So in the spirit of reconciliation, MND Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to the elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. And I'd like to acknowledge the Wiradjuri people on whose land I am and pay my respects to elders past, present and future. So our presenters today are going to tell us about their research into the different aspects of the role and the impact of your genetic makeup on MND and how insights into your genes can improve diagnosis and potentially treatment. Our presenters are Emily McCann from Macquarie University in Sydney, and we have Anthony Akkari from the Perrin Institute in Perth. So Emily will be presenting first and Anthony afterwards. As with our previous sessions, each presenter will talk for about 20 minutes, and then we'll have a joint Q&A session at the end. So if you have any questions to submit, please use the chat or the Q&A function if you're on Zoom, or if you're on Facebook, just use the chat function there, and we'll collate all the questions and put them to the speakers at the end. So as I said, first up, we've got Emily, so please take it away.
Thank you, Gethin. So I'll just share my screen. So hopefully everyone can see that now. So yes, I'm Emily. I'm a postdoctoral research fellow at the Macquarie University Centre for MND Research here in Sydney, where I belong to the genetics and genomics team. And our primary research goal is to discover novel genetic causes of MND. So to start with, I just want to talk about genetic inheritance. So genetic inheritance is the fact that we inherit our genetic material or our DNA from our parents. So parents transmit their genetic material to their offspring. So what you can see in this diagram on your screen at the moment is we have this child who has inherited one copy of this gene from their mother and one copy from their father. So we inherit 50% of our genetic material from each of our parents. So this orange copy of this gene that this child has inherited from their father has a typo in the sequence of DNA letters within this gene. And we refer to this typo as a genetic variant or a genetic change or an alteration or a gene mutation. So when you see this diagram and you think about genetics, your first thought is probably the familial form of MND. And while it's true that most of what we know about the genetics of MND has come from studying the familial form, there is still a significant genetic component to the sporadic form of MND as well. So tonight I'm hoping to show you how we can use genetics to better understand both familial and sporadic MND. So at Macquarie, we have quite a unique approach to our research. So individuals affected by MND first present to Macquarie Health at the neurology clinic where they see Professor Dominic Rowe. And from here, these individuals are recruited into the Neurodegenerative Diseases Biobank, as well as many of their family members. And all of these individuals very generously donate samples to be a part of the biobank. And these samples directly feed into the research that we do at the centre. So as part of the genetics and genomics team, our discoveries are really at the foundational level and really feed directly into all the other research streams going on in our centre. And they also feed directly back to the clinic and can be really informative for the clinicians and the whole clinic team. So to set the scene for everything I'll be talking about tonight, I just wanted to start off with a bit of history of our understanding of MND. So back in the mid-1800s, Charcot first described MND and coined the term amyotrophic lateral sclerosis, which is the internationally recognised name for MND. And then a few years later in 1880, Osler described the first family with a history of MND. So this was quite a large family that came from Vermont in the US, and there were multiple individuals across various generations of this family who were affected by MND. And this gave us the first clue that there was some sort of hereditary or familial component to MND. Then over the next few decades, there was an increase in the awareness and understanding of MND, particularly in terms of the variability. So we know that no two individuals have the same experience with MND, and we see vast differences in the age of onset, in the site of onset of MND symptoms, and also in the rate of progression of MND. So it wasn't until a century or so later when we had our first breakthrough in understanding the biology of MND. And this came with the identification of these hallmark pathological feature of MND, which are these ubiquitinated protein aggregates. So what these aggregates are is essentially clumps of proteins. So under normal conditions, proteins fold into their correct conformation, which allows them to perform their normal biological function. But what happens in MND is that these proteins do not fold correctly, and instead they become sticky and stick together into clumps. And these clumps are what we call the aggregates, and they deposit themselves within effector motor neurons. Then a few years later, in the early 90s, we had our first breakthrough in understanding the genetics of MND with the discovery of the first MND gene called SOD1. So it was found that a mutation within this SOD1 gene was being passed down from generation to generation within that large Vermont family, which I just described, and this was what was causing MND. So it was 113 years between this family first being identified to actually identifying what the cause of MND was within this family. About a decade later, we had a breakthrough in the human genetics research space, and this was the completion of the Human Genome Project. So what the genome is our complete genetic profile of DNA as humans. And this is 3 billion letters of DNA sequence. And in each and every cell of our bodies as humans, we have a copy of this human genome. And as part of the Human Genome Project, this entire 3 billion letters of DNA was sequenced within a single individual. And what this did was essentially give us a roadmap to understanding what these letters were telling our body to do. And ever since it's been completed, the Human Genome Project has acted as a pivotal resource in human genetics research, and MND is no exception. So the year following the completion of this project, we had the identification of the next MND gene called synataxin. So here we had 11 years between the first and second MND gene discoveries. A couple of years later, we had another breakthrough in understanding the biology of MND. And this was directly linked back to these protein aggregates that we just spoke about. So it was identified that the principal component of these aggregates was a particular protein called TDP43. And over the next few years, my team leader or supervisor investigated the gene which encodes for this TDP43 protein, which is a gene called TARBP. And he found that mutations within this gene were being passed down from generation to generation within families and causing MND. So this illustrated a really critical link between the biology and genetics of MND. So here we had 4 years between these MND gene discoveries, and then the next one came quickly the following year with FUS identified. So you can see here that we have this acceleration in our identification of MND genes. And in the same year of 2009, we had the advent of a revolutionary DNA sequencing technology called next generation sequencing. So this gives us the ability to sequence millions of DNA molecules really, really rapidly. So this facilitates the generation of a very large amount of genetic data, which allows us to sequence the entire genome of an individual within just a few days or even just a few hours. So this is in stark contrast to that Human Genome Project, which took 15 years to complete and billions of dollars to do the same thing and sequence those 3 billion letters of DNA in a single individual. So based on this, you can really appreciate that this technology really revolutionized all of genetics research. And again, MND is no exception. So just a few years after we had next generation sequencing available, we had the discovery of the most common known cause of MND, which is a mutation in C9orf72. And in the years following, we had an explosion in the identification of MND genes, so that every year we had at least one, if not a number of different MND genes discovered, and this was all facilitated by that next generation sequencing technology. And here I've really just provided a snapshot, as we have over 30 different MND genes that have been identified so far. And in pink, I've just highlighted those that I've directly been involved with discovering. So with all of these MND genes being identified, we had some understanding of the molecular origins and the biology behind MND. And the first thing that we learned was that MND had shared molecular origins with another neurodegenerative condition called frontotemporal dementia. And the genes showed us that these two conditions actually sit on a spectrum of neurodegeneration rather than each representing separate conditions. So at one end of the spectrum, we have pure MND, and at the other end of the spectrum, we have pure frontotemporal dementia. And mutations within any of these genes could cause pure MND or pure FTD, or in some cases, it could cause symptoms of both conditions in the same individual. Another important thing that MND gene discoveries have taught us about MND is that there are just a few key biological processes implicated in the mechanisms behind MND. So while there are thousands of different biological processes at play in the human body, just 3 particular processes have continuously been implicated in MND by gene discoveries. So MND genes cluster together within these processes. So this highlights that these processes have really important roles in the biology of MND, and also flags these processes as being really important targets for us to develop effective therapeutics for treating MND. So now that I've told you about all these different MND genes and what they've taught us, how much of the cause of MND can these genes actually explain? So here, this is from work that we published a couple of years ago, looking at the prevalence of MND gene mutations in 212 Australian families with a history of MND. So if you focus on the pie chart here, you can see that around 40% of these families carry a mutation in C9orf72, while another 14% of families carry a mutation in that SOD1 gene. And a handful of other families have mutations in some of the other MND genes. So this explains what's causing MND in those families. But in almost 40% of all these families, we still have an unidentified genetic cause of MND. So this highlights a really critical gap in our knowledge of the genetics underlying MND, and it's something that we're working really hard to solve. So now looking at the sporadic form of MND, and again, this is from a paper we published just last year. And here we were looking at 616 Australians who are affected with sporadic MND. So among these individuals, we found that around 9% had a mutation in a known MND gene, and a further 25% of these individuals had some sort of genetic variant that could be contributing to their cause of MND. But this leaves at least 65% of these individuals with an unknown genetic predisposition to MND, again highlighting that we still have a lot to learn about MND genetics. So this brings us to look at the differences between the genetic causes of familial and sporadic MND. So in familial MND, we know that we have many individuals across multiple generations of a family who are affected. So this suggests that genetic changes of very large disease effects are inherited from generation to generation. And this is what we call a mutation. And this is sufficient in isolation to cause MND. But on the other hand, in sporadic MND, we have individuals who have no known other family members who are also affected. And this suggests that there are genetic changes with small disease effects that could be contributing to the cause of MND in these individuals. And this is what we call genetic risk factors. But these genetic risk factors are not sufficient in isolation to cause MND. So the small disease effects contributed by these genetic risk factors need to combine with other similar sized disease effects in order to reach a threshold to trigger the onset of MND. So let's rehash what we do know about MND genetics and what questions that leaves us with to answer. So we know that over 30 MND genes have already been identified, and these genes can explain up to 2/3 of familial MND and up to 10% of sporadic MND. But this still leaves us with up to 90% of all individuals living with MND that have an unknown genetic cause. However, the literature has shown us that 40 to 60% of the risk for developing MND, whether it's familial or sporadic, can be attributed to genetic factors. So there's quite a large disparity between these two numbers. So the big question that we're left with is, what are the unidentified genetic risk factors for MND? So now I'm just going to explain a bit about how we're actually searching for these risk factors. So to start with, let's look at the multi-step hypothesis of MND. So as part of this hypothesis, it's suggested that 6 individual steps or hits are required to accumulate within an individual to trigger the onset of MND. And within these steps, a gene mutation can account for multiple steps. So a TARBP mutation accounts for 3 steps, a C9orf72 mutation accounts for 4 steps, and a mutation in that SOD1 gene accounts for 5 steps. So these genes make MND familial because very few additional steps are required to trigger the onset of MND. So that then leads us to look at the differences between simple and more complex forms of genetic inheritance. So in simple genetic inheritance, we have one mutation within a single gene that causes MND. So an example of this is familial MND, which is caused by a SOD1 mutation being passed down through the generations within a family. On the other hand, looking at complex genetic inheritance, we have multiple genetic risk factors at different locations across the genome, all within a single individual, and all contributing small disease effects that combine together to trigger the onset of sporadic MND. And because there are multiple genetic factors at play here, we call this polygenic inheritance. And as part of that same study that we published last year, we also gave some evidence for the polygenic inheritance to have a role in MND. So again, looking at these 600 individuals, we found that 42 of these individuals carried more than one genetic change that was contributing to their cause of MND. And we found that these particular individuals, who are shown in blue, had a younger age of MND onset compared to the individuals shown in black, who did not have any genetic factors that we could tell were contributing to their MND. So this gave evidence to support that these particular individuals had a high inherent risk for developing MND, and therefore supporting a role for polygenic inheritance in MND. So as I've mentioned, these genetic risk factors have quite small disease effects. So this can make them very tricky for us to identify. So in order to identify these genetic risk factors, we need to look at very large numbers of individuals to try and find out what these risk factors actually are. So in our effort to do this, our research group is involved in a large international consortium called Project MIND, which has the ultimate goal of collecting the genetic profiles of 15,000 individuals affected by MND from across the world. And our Australian team has already contributed genetic profiles from over 600 Australians living with MND. And our role in this consortium was launched back in 2015 by a grant that we received from MND Research Australia, which was funded by generous donations received from the Ice Bucket Challenge, which many of you will remember from a few years ago. So in order to identify these genetic risk factors, we are looking for genetic changes that are more common or more frequent among individuals affected by MND than these genetic changes are in the general population. And by taking approaches similar to this, and as part of Project MIND, our team has been involved in identifying a number of novel MND genes over the last couple of years. And lastly, I just want to touch on a novel approach that I'm taking to MND gene discovery. And this is as part of work of my fellowship, which was generously awarded by MND Research Australia at the end of last year. So most genetic research in MND and in other conditions has focused on small genetic changes. So these are essentially typos in our sequence of DNA letters that affect a single letter of DNA, such as this T to G change that you can see. And if we think about this in a sentence, this is similar to changing a sentence from saying the cat is fit to changing the sentence to the cat is fat. So you can see that we've simply changed one letter within this sentence, but it's completely changed the meaning of what that sentence is telling us. But what I'm starting to look at now is these more complex and larger genomic changes that, instead of affecting just a single letter of DNA sequence, are rather affecting long stretches of many letters of DNA. So hundreds or even thousands of letters of DNA in a row. So in this example here, you can see that this red chunk of DNA is completely removed from this DNA sequence. So thousands of letters of DNA have been deleted. So if we think about this in terms of a sentence again, we have our original sentence saying the boy was tall and his sister was very short. And we have removed all of these letters and words shown in pink here, and the sentence now becomes the boy was short. So you can see that this has had an even more drastic effect on the meaning of our sentence. And the sister has been completely removed from existence, and the boy has changed from being tall to now being short. And we see similar drastic changes in what our DNA tells our bodies to do when we have these really large genomic changes. And we think that these large changes could also be having a role in MND. So to wrap up, I just wanted to go over some of the benefits that our gene discovery has for patients. So first, in the clinic setting, our discoveries directly translate to diagnostic testing for patients. So this means that we can provide an answer for what is causing MND in quite a number of patients. And this in turn can facilitate early diagnosis of MND and allow clinicians to provide the most appropriate clinical management strategies for these individuals and hopefully delay the progression of MND. Our discoveries also have benefits for genetic counselling. So family members may wish to undergo genetic testing to identify whether or not they carry a mutation that could cause MND. And as part of genetic counselling, they can make informed decisions about whether or not to undergo this testing, bearing in mind the benefits and burdens of finding out the result in either scenario. Our genetic discoveries can also be used in pre-implantation genetic diagnosis coupled with IVF to prevent the inheritance of these MND-causing mutations in future generations. And importantly, this process can be performed blindly so that the individuals undergoing the testing do not need to know whether or not they themselves carry a mutation that will cause MND, but can still have the peace of mind that their future offspring will not have these mutations. So in the future, we're hoping to work towards more personalized medicine approaches for treating MND. And we're hoping that we'll be able to genetically profile individuals living with MND to first predict the rate of progression, but to also hopefully one day identify what the most beneficial therapeutic for each individual will be based on their genetic profile. But of course, we still need to identify these therapeutics first. So lastly, that just leaves me with a number of thank yous. So firstly, to my research team for your support day in and day out. And of course, to the funding bodies, because we couldn't do our research without the money that you provide to us. And in particular to MND Research Australia, who has been a long-standing supporter of our team's research. And of course, to the wider MND community and for your participation in our research, because again, we wouldn't be able to do what we do without you participating and supporting us. And lastly, thank you to everyone for listening and coming along tonight. I'm happy to answer any questions in the Q&A session later on. So that just leaves me to hand over to Professor Anthony Akkari from the Parent Institute in Perth to talk about the translation of genetics into therapeutic development for MND.
Hi, can everybody see me and hear me? I'll take the silence as yes. Good evening and good afternoon, everybody. As has been said, my name's Anthony Akkari. I'm here to talk to you about our MND group over here in Perth, Western Australia, and what we're doing in MND genetics and genetic markers and therapies. Emily, thanks very much for a wonderful presentation that really allows me to explain what we want to do with those genetic markers and how we want to try and translate those into therapies. Just a little bit about my background. I'm from the pharmaceutical and drug development industry, and I've been working in the US for quite some time. And I returned to Perth about 4 years ago to set up this group as a research professor at Murdoch University and at the Perrin Institute as the head of motor neurone disease genetics and therapeutics. My primary role over the few years, over the 15 years that I was in the US and ongoing in my engagement with industry, has been to integrate those genetic discoveries that Emily talked about into the drug development pipeline to come up with these personalised and stratified therapeutics. You'll hear a little bit more about a couple of companies, and specifically Black Swan Pharmaceuticals, who I'm a founder and a CSO of, a little bit later on. And I also work with Genius, which is an ALS-specific or motor neurone disease-specific company. Both companies have been set up specifically to try to translate therapeutics in motor neurone disease and genetics as well. I think what's super important for me to just mention at this stage of the presentation is that the Perrin Institute, which was at the time prior to my leaving, was the Australian Neuromuscular Research Institute, and a couple of colleagues of mine, those being Professors Steve Wilton and Sue Fletcher, began working on antisense oligonucleotide therapeutics. You may be familiar with them from a lot of the press in industry, at the moment with vaccines and so on. And these are more of your RNA analog therapeutics. In other words, they directly target genes and they modulate gene expression. Steve Wilton and Sue Fletcher had been working on this at the Perrin Institute for quite some time, and I'd always maintained a link with them whilst I was in industry. And over those years from the early 90s through to 2016, they hit a milestone, and that was that these antisense therapeutics that they developed for Duchenne muscular dystrophy, one of the first ones in 2016 made it to through clinical trial development and then to patients. So that was a real milestone with them, with their FDA approval for that therapy to go into patients. And that had been capturing my attention for some time and my imagination, because me and others think that antisense therapeutics, which are these very precise therapies that modulate the genes that are being discovered, are really going to make a big difference for at least a subset of MND patients. And we don't know how many or what big that subset is, but we are fairly confident that that's going to make a difference. And so it was on the basis of those discoveries that I was willing to come back and combine this, the genetics tools that I've learned to use in industry in combination with these antisense therapies to try and come up with solutions for motor neurone disease. Just briefly going back to the discovery of a number of these genes. Also, the group that I was involved in here in the early 90s, which was that of Professor Nigel Lang, was a collaborator with those that discovered this first SOD1 gene. And so at that time, when I was working in the neurogenetics lab of Professor Nigel Lang, I became both involved in the research in motor neurone disease, but also my father was diagnosed at that time with motor neurone disease as well. So in one or a few years there that spanned that period of genetic discovery, became both a researcher in that space and also somebody who was about to embark on a journey with my father suffering from motor neurone disease. I think both of those things, my genuine interest in trying to understand what's happening here, in addition to that experience, continue to maintain my passion throughout the years for understanding this disease. This is the MND group. We're a small group. We were formed in about 2018. The two postdocs that we have here, Lauren Flynn and Oliver, are embedded in the laboratory of Professor Steve Wilton. And it's because of that embedding in that group that we've been able to very rapidly develop a pipeline of therapeutics that we're looking to drive to translation. And I'll talk to you a little bit about that. And then you can see here we have our 4 PhD students, our research assistants, and honour students as well. And I report into all of these folks, as you could imagine. So this audience is well aware of a number of the significant problems for MND. But the problem that we're focused on is that it's without an effective therapeutic. And without going into all of the numbers here, currently in Australia, there's only one MND therapeutic approved with a limited effect in some patients, and that's riluzole with a 3-month slowing of progression. So we and others think that a limited understanding of the disease mechanisms in motor neurone disease has had a limited clinical trial success. And over the last decade alone, particularly for motor neurone disease, there have been greater than 20 MND clinical trial failures. This is not peculiar to motor neurone diseases, more broadly neurodegenerative diseases. And we think that it's important to develop the genetic markers to improve the detection of patients that might respond to these drugs. In other words, the genetic markers that might underpin their mechanisms and help us better classify those patients. And so our approach here is to bring the co-development of genetic markers with targeted antisense therapeutics, or RNA analog drugs. And I'll use those words interchangeably, but essentially we refer to them as antisense therapeutics. So we want to bring those two together and co-develop them preclinically, so that when we receive our approvals to go into phase 1, that they're there ready for both stratification using a genetic marker or enrichment, and we have our precisely targeted therapies. So our MND Genetics and Therapeutics Lab, based here at the Perrin Institute, has 2 main goals. As I said, to work on those genetic markers or DNA markers that identify the disease mechanisms, that they contribute to that risk that Emily very nicely explained, that contribute to the disease course, and again, help us understand some of the underpinning mechanisms. And with those data, to develop our antisense therapeutics that precisely target those disease mechanisms. So an integration of both of those things together. And that's going to be the focus of the rest of the discussion today. So just a few slides on MND genetics. I'd just like you to focus on the sporadic ALS circle here that you can see. And as Emily had mentioned, 90% of the sporadic motor neurone disease cases have no known mutations. So we need to identify more of these markers so that we can understand a bit more about the mechanism in a subset of these patients. And that can help us enrich in clinical trials for those patients, and also help us better target those patients with our therapeutics by understanding their mechanism. So if we have those kinds of markers, what can we do with them? Well, the idea of having those markers, those genetic markers, is so that we can change the outcome of a clinical trial. So if you look to the left here, you'll see a group of patients that have just completed a clinical trial. And we've seen a mixed response to the drug, which is the usual scenario for a therapeutic. Now, if, for example, you had 10 individuals that responded to therapy and 90 that didn't, all up 100 people in the trial, you then compared the 100 on average for their response against placebo. The effect is going to be washed out by the 10 people for whom we did see a response, because we have no way of picking those individuals out from the rest. But what if we had a genetic marker that would enable us to stratify them into these groups, into a genetically defined responder group, into a non-responder group, and as you'll be aware from the news media and COVID, an adverse event group. Well, most importantly, and for the focus of this talk, we could focus our development strategy towards the genetically defined responder group. And that's the essence of what we want to do with genetic markers that might identify a disease mechanism for patients. So there are multiple motor neurone disease mechanisms that have been presented in the literature. We have a small group. We can't target everything with our genetic marker approach. We only have limited resources in that respect. But also we can't target everything for our antisense therapeutic approaches. But essentially we're focused on protein aggregations, those effects that may disrupt the function of the axon, the part of the neuron that conducts transport back and forth and repair and healing, and also potentially the mitochondrial dysfunction aspect of the neuron, which is the power management system. And so they're the large focus areas of our small group. So in terms of genetic markers, we have made some progress. We've identified a number of these genetic markers that are called structural variants that you heard about earlier. A number of them, the structural variants have been not well examined in the prior 20 years with our high throughput technologies. And you need to take a bit more of a custom approach to certain kinds of structural variants. And that's the approach that we're taking, which is a little bit more laborious, but is beginning to yield us some understanding. So these are the broad areas of focus for our structural variant approach. That is in the toxic protein buildup, the nerve transport and signaling, mitochondrial function or mitochondrial dysfunction, neuroinflammation, and potentially neuroprotection. Our approach to this is to move through a candidate selection, assay development, all the way through to a retrospective analysis in clinical trials. And right now we're talking to our potential collaborators across the world to see how we can better access clinical trials or actually currently being undertaken or that have been completed for which we can get DNA samples to begin to look and see with some of these markers whether we can stratify and identify responder populations. Intriguingly, in 2017, a group in Europe with the lead author Van Eck did in fact show that a subgroup of patients that carry a mutation in a particular gene called UNC13 responded to a lithium-ion treatment. Prior to the use of that genetic marker to identify that group, all studies reported a negative finding for those clinical trials. So the proof of concept is out there in the literature, and we're pretty confident that moving forward that genetic markers that stratify patients are really going to help us, particularly in the MND space, and that's already been shown with the Van Eck work that was done. In lockstep with that and in co-development, in those broad areas of toxic protein buildup, nerve transport and signaling, and mitochondrial dysfunction, we're also developing antisense therapies. These are therapies that precisely target disease genes, disease-causing genes and mechanisms. And we have a number of those here, and we've been able to do that very effectively because, as I mentioned, we've been embedded in the group whose core capability is that. For one of our genes in particular, for SOD1, where we have an antisense therapeutic developed, we have received some funding both from MND Australia and from VitaMND, and we are grateful for the support there. But one of the big problems that we have is that as we develop these therapies, to try to get them out of our laboratories' preclinical activities into what we call phase 1 readiness, there's a little bit of a funding shortfall. It's difficult to engage with partners to fund that next stage of toxicology studies so that we can engage the FDA and other regulatory agencies to allow us to translate those into readiness for phase 1. And so in response to that, we formed the small pharmaceutical company called Black Swan Pharmaceuticals. This is a purpose-built company to translate the therapies from our lab to make them IND submission ready for phase 1 clinical trials. We're hoping that as we develop these therapies and we can secure funding for this startup, that we can de-risk our therapies at the early stage and attract the support of biotechnology and larger pharmaceutical partnering companies. Black Swan was formed because of my connections back into the US pharmaceutical industry, and we assembled a group of people who've got enormous strength and capability in taking therapies from the lab and getting them phase 1 submission ready with the regulatory agencies. And so the team that forms Black Swan has overseen that process for over 180 small molecules, and we're now moving into the antisense oligonucleotide space. And it's our hope that that will be a translation engine for these therapies, providing that opportunity to have more shots on goal for phase 1 clinical trials and beyond. So what does this mean for patients? Genetic markers improve the chances of clinical trial success. We can enrich for disease mechanisms, and we have a greater chance of stratifying responder subgroups. The antisense therapies offer us a rapid development opportunity and allow us to precisely target genes based on our improved understanding of MND gene function or disease mechanism. We are a small group, but because these antisense therapies are rapid to develop, we have 10 in our pipeline. We have a better chance of targeting specific MND mechanisms with these, and they have begun to demonstrate that they are a safer and quicker way to develop therapeutics than conventional therapies. But this certainly, I just want to be clear, they are not the only answer to motor neuron disease therapies. They are part of a larger toolkit of therapeutics. We think that, as I said from the beginning, a subset of patients will work well with these antisense therapies. So in terms of the state of play, an update from the Food and Drug Administration, the FDA has recognised the importance of rapid development of these antisense or RNA therapeutics for diseases, for rare diseases. And they've come up with a specific guidance, which is very important for researchers like myself and others around the world who are looking at these antisense and even other small molecules. This will, the FDA has very succinctly set up a toxicology guideline for getting these therapies in and through the FDA, which should, with hope, streamline getting them ready to go into patients for personalised therapies. I just want to be clear here, this is not the answer to everything. We still have to pursue a responsible development strategy in that all toxicology work has to be done. And whilst this will help us streamline therapies, we still have to take all of the necessary steps to ensure safety, which is our highest priority. But we do think that for rare single point mutation forms of motor neuron disease, that this guidance will be essential and important, but it could also be more broadly applicable to sporadic forms of motor neuron disease. I want to take the time to thank the families that are in the people that have donated their DNA to our research and motor neuron disease patients from around the world. There's a lot of institutions for us to thank and all of our collaborators, but our funders we cherish dearly and thank them very much for their support and faith in the work that we're doing. Thank you.
Great. Thank you, Anthony, and thank you to Emily. That was a fantastic overview of genetics, right from how they might cause MND to how we can target these genetic changes to actually provide some treatment options. So we've got a few questions up there. So Emily, if you could come back online as well, please. Great, thank you. So first up, we've got a few questions lined up. So first question is, are the treatments of the gene-related treatments quite consistent worldwide, or are some countries a bit further ahead than others?
So I'm taking that to mean, what are the treatment options for people with motor neuron disease mutations? If that's the answer to the question, and I think it's pretty consistent across the world. We have riluzole here. I know in the US they have another drug as well called edavarone. There's a lot of skepticism about the value of that particular drug. It's also incredibly expensive with almost no benefit to the best of my knowledge. But essentially, the treatment options are the same for mutation positive patients as they are for those with no known mutation. I will add that there is one exciting antisense therapy called tofersen, which is in phase 3, and I hold great hopes for the approval of that. That is a drug that targets the SOD1 gene, and it is for SOD1 mutation positive patients, which is about 1 to 2% of the MND population. So that's a gene-specific therapy that it is my hope will become available once the phase 3 studies have completed.
Okay, thank you. So another question we have here is, has there been any genetic work done in people who have are more slowly progressing versus fast progressors? Have there any genetic associations been identified with those two different sort of disease outcomes? Perhaps Emily, would that be more in your...
Did you want to speak to that?
Yeah, I can take that one. So to date, there's work going on in that field, looking for what we call genetic modifiers of the presentation of symptoms of MND. But to date, there hasn't been any particular genes or variants that have been associated with that. But there are suggestions in the literature that particular mutations and genes that we know that cause MND generally associate with having particular symptoms or rates of progression associated with them too, but they're very generalized and there's no hard and fast rule between those yet.
So just to add to that, we very, very recently published a structural variant which is associated with a more rapid rate of decline in the Stathman gene. That data is very new. It needs to be validated by other groups, and that's what we're trying to do right now. But I think there's some efforts along those lines as well. So that was very new, Emily. You probably didn't even know about that yet. We just got it. Don't expect a lot of people to know about it at this point.
Okay, thanks. I mean, another question here is, if you have patients which have the same mutation, do you see a lot of difference between the patients progressing even though they may have the same mutation? Do you get a uniform disease with each mutation, or is there a lot of variation?
Yeah, I can speak to this one again. So yeah, that's a good question. Again, similar to the last question, we do see some general associations between the mutation and rates of progression. But again, there's no hard and fast rule. We see patients within the same families that carry the same mutation, and some of them will live with MND for very many years, and others will have very rapidly progressing MND. So there is still a lot of variation out there with the symptoms, even with the same genetic predisposition.
Okay, and with sort of perhaps we'll target a bit more at Anthony, with a number of sort of gene therapy trials now coming online, how's the best way for patients in Australia to keep track of those clinical trials and ideally become involved in those clinical trials when they finally get to Australia? Any suggestions?
It's a great question. So clinicaltrials.gov, which is the US clinical trials registry. You can look on that, and that has clinical trial information. There are also the ALS organisations, particularly in the US and Europe, that advertise the clinical trials that are happening. In terms of them, when you have sites here in Australia, I guess the best way to participate in those is to talk to your neurologist to find out what clinical trials are happening and whether they'll be suitable or not for them. That's my best explanation. I don't know, Emily, have any additional advice there?
No, I would completely agree.
Yeah, and I think just a small addition to that is it's probably most likely that only the phase 3 trials at the moment would be coming to Australia. I think the phase 1-2 trials, where they're testing some of the other genes beyond SOD1, such as C9orf and FUS, they are the phase 3 will come to Australia. So if I talk to your neurologist and make sure that you are in the national registry, and possibly hopefully then as well in the SALSA database, because then your genetic data will be to hand, and when they can identify patients that will be suitable for these trials very quickly through that registry and through those databases. So if you if you're not already within the registry as a patient, then I would recommend you talk to your neurologist to become part of that registry, because then the data is there and people can be identified very quickly. And I'd also say that to Bruce as well, who gave us a question through the chat. Again, do we want your DNA? The researchers are always welcome everyone's DNA who's involved with MND, and if you get yourself into that registry, again, your DNA will be then available, and we can find out about the best treatment options. Another question we have now is with I think Emily, you were saying that some people have quite complex genetic makeups and perhaps multiple genetic associations with the disease. If perhaps through Anthony's research we have a number of different genetic treatments, could people try multiple gene therapies, do you think, together to target several genes at the same time?
I think at the moment, when we think about there being multiple genetic factors at play within an individual, each of those is only contributing such a small disease risk. So I don't think we would know exactly how much of each to target just at this stage. So I think there's a lot more research for us to do to find out how big these effects are and how they're working before we can get to the point where we can try using multiple gene therapies to target all of them at once.
And on a slightly related question, you were saying quite often there will be multiple genes in the same pathway. And say at the moment we have SOD1 is being tested, and hopefully we'll get some positive results. If you have other genes in that pathway, do you think it'd be possible to use SOD1 to target the whole pathway rather than perhaps just people with SOD1 mutations?
Yeah, so Anthony might be able to add to this as well. But yeah,
of course.
My thoughts would be you would have to look at the pathway itself and figure out which gene or which product from which gene was having the first effect, and then you would be able to target that one and hopefully stop the cascade from moving down the process or pathway.
Yeah, so I appreciate Emily's comment. I think we won't be saying anything too dissimilar. There's science is a contact sport, for want of a better term. SOD1, misfolded SOD1 is observed in a subset of ALS sporadic patients. There's a thought there that an antisense therapy that could knock down SOD1 might affect those aggregates. That's at least one hypothesis that we're working on, and I hope that Ionis and Biogen would be looking at that as well. I think we have to test that idea and see that in patients, do we have evidence that by knocking down SOD1 in sporadic patients through animal models and through in vitro work, could we get some efficacy readout that we could then give us a weight of evidence to take that therapy into sporadic patients? We have to proceed here with asking the question, but also creating the evidence that allows us to do this. We shouldn't just rush in and do those, but that's what we're doing. We're actually testing that. And I'll come back to the idea of combination therapies. Again, I agree with Emily, and I think that for example, something like TDP-43, there's a lot of discussion around whether directly targeting TDP-43 would even be viable. We're certainly working on the possibility that it might be under the right set of conditions. But also there are various factors that TDP-43 regulates. It regulates over 6,000 messages is my recollection. And one of those is important in nerve growth and repair. And so potentially thinking about upregulating that and downregulating that feed-forward mechanism in TDP-43. That's very experimental and quite a bit away, but we are thinking about it.
Thank you, Anthony. I have a question here from Duncan Bailey, a regular contributor. Doesn't stratification increase the possibility of a false positive? How can you accommodate that?
I'm not sure what that means. With every analysis that you do, there's always the risk of a false positive or a false negative result. I guess the best way to accommodate that is in a number of ways. That is, prospectively state your hypothesis and lock that down going into a clinical trial. So if you have a prospectively locked down secondary objective in a clinical trial, that minimises your degrees of freedom, your opportunity to, fudge your interpretation, for want of a better term. I think the best possibility is that you do a discovery stratification in your first clinical trial. You identify that population, and then you test that in the second clinical trial, and that gives you a second verification of that stratification. If that answers the question, I'm not sure, but I hope I didn't go off too far.
No, I think that's probably about right. I think it's probably the risk of when you stratify, you get very low numbers in your population. So perhaps by doing an independent verification, that will hopefully help to avoid some of those false positives. We also have a couple of questions really around genetic testing and what sort of advice we can offer around that. Obviously, none of us are genetic counsellors, so really about what's the best practice if you have carriers in your family and what can you do going forward. I'd recommend, if you do carry the genes in your family, that you talk to a genetic counsellor and get advice before testing as well as post-testing if you decide to go forward. I don't know if either of you two would offer any further advice beyond that. Perhaps Emily, you probably deal with a lot of people obviously in this situation.
Yeah, so I would say the exact same thing. So at the Macquarie Clinic, we have a genetics counsellor who sees family members and patients as well. And she actually gave a webinar a couple of weeks ago, which I don't think the recording is available yet, but that could also be a really nice resource for people to visit if they're interested.
Yeah, I think it's a very strong message to make sure that you talk to genetic counsellors. Those guys are fantastic and can explain the ins and outs, consequences, and your options, and lay it right out on the table for you to make it very clear. And I think we're getting towards the end of our question. We also had another question here about gut health and MND, which isn't quite related to genetics. So there is quite a lot of work being done at the moment in the microbiome, basically the genomic makeup of your gut. But there certainly is some work being done out there at the moment to look at how gut health might affect MND. I think there isn't any unequivocal evidence saying one way or the other. I think there's definitely indications that it can affect it, but I can't say that I think we've got to the point where we can say exactly how variations in your gut health will affect MND. I don't know if Anthony or Emily have any know a bit more about that than I do.
I'm sorry, I don't. I know that there's growing interest over here too at the Parent Institute, but it's not something that I'm located. It's way out of my wheelhouse.
Yeah, we did fund Professor Pamiga up in Queensland actually to do a study in the gut microbiome this year. So we'll hopefully see some results coming out of that in the next few months actually. So I think that takes us pretty much to the end of all our questions, and I'd just like to thank both of you again for an absolutely fantastic overview, and again explaining some very complex concepts very simply so we can all understand, especially that 6-step. That was fantastic. I understand a lot more about that 6-step process now. Thank you, Emily, because I really struggled with it before. So going forward, we have the next seminar, the next session will be the 15th of June, and we will have 2 researchers discussing the role of the immune system in MND. We're really looking forward to that, as that's a very topical issue at the moment. So please, everyone who registered will receive a link to the recording, and it'll also be up on our YouTube page for those that you missed it. And please distribute that to your friends and networks as well. We like to get these out as widely as possible, especially given the quality of the presenters. So please, so good night. Enjoy the rest of your evening, and I also hope you all get to appreciate some of the other events occurring around Australia for MND week. So good night. Thank you.
Good night. Thank you.
Thanks.